Um, Moving on to our next speaker, he's my mentor, is where I have started with functional medicine from. His topic for today is methylation and chronic disease. He is Dr. ANISH MUSA. He is a renowned functional medicine practitioner who possesses a wealth of knowledge and experience. I'm delighted to introduce Dr. ANISH MUSA, an accomplished ophthalmologist with an impressive 25-year journey in the field of ophthalmology. He is a co-founder of IAFM, coupled with a certification in functional medicine from esteemed Institute for Functional Medicine, USA. Dr. Musa serves as the guiding light at Ultra Wellness Clinic, a pioneer institution that specializes in an array of health concerns, including metabolic syndrome, hormone health, mold toxicities, gut disorders, and autoimmune conditions. Drawing from the powerful principles of functional medicine, he highlights the importance of personalized care. His dedication has led to the creation of functional lab analysis software, which is a game changer in functional medicine. This groundbreaking tool translates lab reports into optimal ranges, providing practitioners worldwide with a comprehensive diagnostic approach. I'll repeat, his topic for today is methylation and chronic disorder. Over to you, sir. Please enlighten us with the topic. Yeah. We cannot hear you. Can you please unmute yourself?
Thank you very much, Dr. Karishma, for introduction. And hey guys, today's my topic is on methylation. So before I can deep dive in methylation, already you heard a lot about the homocysteine and methylation and functional medicine. So my real objective is to give you idea about functional medicine through this methylation process. So here I would like to share my screen. And Dr. Karishma, everybody can see my screen, right?
We can see your screen. Can you please maximize?
Yeah, can you see it?
Clear, visible,
Okay.
and you're loud and clear as well. Please go ahead.
Okay, so let's talk about the methylation. You heard a lot with Dr. Samantha is talking about the methylation is anti-aging. You heard a lot about homocysteine, that we have to reduce the homocysteine. So let's deep dive in methylation. Maybe your patient or you suffering with any kind of cardiovascular disease, maybe it's a myocardial infarction, atherosclerosis, or maybe it's a clotting or thromboembolism. Maybe your patient is suffering with an hormone dysfunction. You heard a lot about estrogen dominance. You a lot heard about the hypothyroidism, autoimmunity. So maybe you or your patient is suffering with an hormone imbalance. Maybe your patient is suffering with an autoimmunity. Chronic inflammation. You heard a lot in functional medicine that chronic inflammation leads to many chronic disease, and that is a basic underlying mechanism that leads to oxidative stress and so on and on. Maybe you are suffering with any kind of chronic degenerative neurological disorder like Parkinson's, Alzheimer, depression, anxiety. Maybe somebody is suffering with an chronic pain, fibromyalgia, fatigue, metabolic syndrome, hypertension, diabetes, and so on. After working with thousands of patients of all this different chronic condition, I came to realize that we have to change our thinking. In conventional medicine, we are thinking very downstream. Our vision is very narrow. So when I was practicing conventional medicine, when I look at the myocardial infarction, I only focus on heart and heart physiology. I don't look at the immune system. I don't look at the nutritional status, or I don't look at the hormone profile. But when I started learning functional medicine, started doing and practice of functional medicine, soon I realized that we have to go beyond that thinking. We have to go upstream and have to see what is happening in the cells. When I talk about the upstream, means I'm talking about what is happening in cellular physiology, what happened at the cell. So by doing an functional study, I came to know for all this condition, there is a one central system that is responsible that we have to understand. So basic physiology or basic understanding of chronic disease is our interaction with our environment. Let's give you simple fundamental. Let's our environmental choices. So if we are exposed to any kind of infection, or we are exposed to any kind of toxins, or maybe it's our bad lifestyle choices, so that's the environment, and that environment affect our genes. and that genes express their functions. And that is what we learn in last 1.5 days about the epigenetics. So that's the expression of a genetics. And depending upon person to person, depending upon their unique genetic profile, they express the disease or dysfunction. Maybe it's an endocrine dysfunction, maybe it's a immune dysfunction or neurotransmitter abnormality or digestion or metabolism, and you have a lot of these things in last 1.5 days. So what I'm going to say is, you can turn on and you can turn off your genetics. Let's say your genetic is exposed and your disease because of some kind of expression of genetics. With the change of environmental factor, you can turn off the genetics. And that central cellular process that you heard a lot is methylation. So I will deep dive in that methylation. It's a very important for so many conditions. So that methylation process I would like to show you here. So what is methylation? So if you look at the biochemistry of methylation, then it's a carbon that is attached to 3 hydrogen molecules, and that is we call CS3. So methylation means in our cellular process, we juggle this CS3 group from one chemical compound to another chemical compound, and that enzymatic reaction happen in each and every second in our cell. So if you want to do any kind of detoxification, let's say pathway 2, so you need methylation to detoxification of many chemicals. And as you heard, all the pharmaceutical, all the chemicals, they are fat soluble. They need to detoxify where you need methylation to be complete. You need CS3 group to attach to that chemical compound, so your body can convert that compound, primary compound, into a water soluble compound. You need for hormone production and detoxification. You heard a lot about the estrogen dominance, but do you know how body can detoxify estrogen? You need methyl donor to convert your E2 into 2OH pathway. So if your deficiency in methyl donor, or if your methylation process is not proper, you cannot metabolize estrogen in safer 2OH pathway. Instead, you increase 4OH in your bloodstream and urine. So that 4OH make you more prone for estrogen dominant cancer. So that's why you require methylation. You require methylation for neurotransmitter, serotonin, and dopamine. You require that methylation for your DNA. And as I said you earlier, that your DNA required methylation to either suppress the genetics or to express the genetics. So let's say if you're unhealthy genes, and if you want to suppress that one, then you require methylation. If you want to upregulate your genes, then you require methylation. So methylation and DNA methylation is actually epigenetic factor that has to be optimized in each and every cell. We required methylation for energy production. You heard a lot about energy production, L-carnitine, Krebs cycle, TCA cycle, where you see B12, folate, and all the amino acids they're juggling, and they are making the pathway to create ATP. So you need proper CS3 or methylation in each and every cell for your energy production. The immune system need methylation. So if you want to fight against the bacteria, if you want to fight against the virus, your immune system read that CS3 group readily available in your lymphocyte. One of the important thing I learned today, that methylation also required for your cell membrane. So maybe you heard that the arachidonic acid is created from the cell membrane, but also phosphodiol E, PE, is converted to phosphodiol serine with the help of methylation cycle and methylation enzymes. So if you have proper methylation, your cell membrane would be perfect. And as you look at the cell, what is the basic function of cell and why this methylation is connected to many organs? Because all the organs, if you look at from conventional point of view, then every organ is different. But from functional medicine lens, we consider that all the cell have some basic mechanism. They require energy, they require methylation, and depending upon that genetic structure of that cell, they express their work. So we have a central process of methylation. And now I would like to go you to teach you about the biochemistry, but don't worry, I will not go and make you hate the biochemistry, because I love biochemistry and I want to specifically show you simple one pathway of methylation so you can understand all methylation biochemistry. So if you look at the one carbon metabolism, that's called a one carbon metabolism, and in center you can see homocysteine. Now this homocysteine is actually an amino acid, and that homocysteine has to convert into methionine. And by the way, what is methionine? Methionine is actually a methyl homocysteine. So it's a homocysteine where the methyl group attached to homocysteine, then it converts that into methionine, and that's the process. And this process required one enzyme. We call this 5-tetrahydromethylfolate. So if you need methyl donor, so methyl donor come in the cell, and the CS3 group attached to this homocysteine with the help of that enzyme, and that convert to methionine. And again, methionine is converted to semi, and then all the cell process go away. Methionine also is an amino acid, and we need that from the food. So if you want to increase the methyl donor from your food, then we have to increase and correct your protein in a sense of methionine. Now you can also see in this slide that homocysteine is also converted to cysteine. And with earlier speaker, you heard about the cysteine and glycine. And the cysteine and glycine is required for glutathione. And glutathione, as you all know, is our primary antioxidant in the cell. And for many processes, we required glutathione. So homocysteine should be optimal in the cell. So let's just assume that cell is working it as optimal capacity. So what is happening? Homocysteine is created, homocysteine convert to methyl homocysteine converts to cysteine, and then you make a glutathione, and that level is down. But just think, if you have a methylation pathway defect and you cannot convert your homocysteine to cysteine, or you cannot convert your homocysteine to methionine, what will happen? The homocysteine level will be increased in the cell, and then from the cell it's going to leak into the serum, where you can check and you can see the level of homocysteine. So in short, this is a central methylation process. Apart from this simple process, there are a lot of other biochemical process that is happening in your brain, that is happening in your liver. But this central biochemistry is very important to understand and make you understand why we need folate, why we need B12, and why we need to check the homocysteine level. Again, was one very important thing I told you, that we required methyl donor to convert the homocysteine to methyl homocysteine. But really, one question I would like to ask you, all the audience, that you know the folic acid, because all the doctor and conventional doctor, they prescribe folic acid. Folic acid is really available, and folic acid is cheap, that you can take and increase your folic acid level. But is folic acid is vitamin B9? What is vitamin B9? So let me give you a simple chemical pathway. When you take a folic acid, let's say you take a folic acid tablet, but that folic acid is not vitamin B9. It's a synthetic version of vitamin B9. I will tell you what is the active vitamin B9. So folic acid has to convert into dihydrofolate. And this dihydrofolate again converts to tetrahydrofolate. And by the way, dietary folate is actually a tetrahydrofolate. And again, this tetrahydrofolate has to convert into 5-tetrahydromethylfolate with the help of MTHFR enzyme. And if you can see closely, then we require two cofactors for MTHFR to convert the folic acid. The active folate is B6 and B9. So let's say if you take a if your patient is having deficiency of B12 and a folate, and if you prescribe folic acid, what will happen? You increase the inactive folic acid in the serum and in the cell. But at the same time, activation of that folic acid to active folate required B12, so your B12 level goes down. So you increase the folic acid, same time you have a deficiency of B12. So when you supplement any B vitamin, understand that B vitamins work in a combination. They work in a synergy. So make sure you prescribe B12 and folate if your patient is deficient in these things. And real question is how we can diagnose methylation defect, and that you heard about the homocysteine level. And here I would like to show you optimal range of homocysteine, because you see in conventional lab the range varies from 15, and even certain lab also 30 homocysteine is considered to be a normal. But here we need to understand the functions of the body. So what we need to understand is every parameter should be in optimal range. So homocysteine optimal range is 5 to 7. So if your level is more than 7, I consider it is not optimal. And as you see that each and every cell required required methylation. So homocysteine is an indicator of poor methylation. So when you want to check your methylation, if you are working on anti-aging, make sure your homocysteine level should be less than 7. Ideally, it should be 5 to 6. The other indicator of poor methylation is, as I said you, we need methyl donor. Why we need B12? Why we need folate? Because they are primary methyl donor. So they have a methyl group attached, and the B12 level here, you can see the B12 level is normal as per conventional lab, but we can say it's below optimal. Even folic acid you can see is below optimal. So here I would like to point out is the homocysteine level is around 14. At the same time, you can see B12 and folate is low. So here we can see methylation, poor methylation is most probably because of dietary deficiency of methyl donor. So in this kind of patient, if you correct the dietary folate, maybe you increase the methionine, you increase the B12, you increase the folate, and where you're going to decrease the homocysteine, and you can optimize methylation. But in this case, you can see the homocysteine level is more than 20. It's a 29. And even you can see the B12 and folate is nearly optimal. So in this case, I would like to really suspect that this patient is having some kind of genetic variation. And that genetic variation, as we know, it is a MTHFR genetic variation. In India, 40 to 50% population have genetic defect in MTHFR genes. So let me explain you what is MTHFR genetics. And if you have genetics, then always keep your B12 folate high. So there are two genes involved for MTHFR. One is called C677T, and that is more clinically relevant. And if you have a defect in this genetics, then it is clinically significant because we have to work on that. So there either it could be a homozygous or heterozygous. Homozygous means both the copies are defective, that we consider as a homozygous. If the one copy of that genes is affected, we consider at a heterozygous. So in this patient, you can see C677T is heterozygous, means one copy is affected. The other genes is wild variety, means it's normal. So in this patient, out of 4 genes, one gene is involved, and that is a defective. So this patient having a mild methylation defect. So if you correct and increase dietary methyl donor, then they will correct their methylation cycle. Now, another patient you can see they have a defect in A1298C, and that is less clinically significant, but it also play a role in methylation. So here is why we need to test this MTHFR defect. As we see that MTHFR is actually an enzyme that responsible for creating a process, that responsible for creating the inactive folic acid to active folate, and that's why it's very important. So if some patient or if anybody have a methylation defect, then their optimal level of B12 and folate is not the same as somebody who doesn't have a defect. So if somebody doesn't have a defect, they can manage their B12 requirement with an 30, 300, or maybe that folic acid is 13 or 14, that is fine for that particular patient. But if you have a genetic defect, then your level should be high. And always you have to consider that you should take an active form of vitamin B12 and B9, that is 5 tetrahydromethyl folate. So if you take an inactive or folic acid, that doesn't going to convert because you have a MTHFR defect. So make sure you understand methylation defect in a detail, so you can advise to your patients in a proper way. Now we had learned methylation is very important. We had learned that methylation is a central process in each and every cell. We learned that how we can diagnose and how we can optimize, how we can diagnose the methylation defect by doing an homocysteine or MTHFR genetics. Now the real question is how we can optimize this methylation pathway. So as you see in biochemistry, we need methyl donor. So when we think about correcting any kind of cellular process or any kind of disease process, we always start from the food. So let me give you idea about what you can do that optimize methylation in your food. Again, what we learn is we need to increase methyl donor, and methyl donor means CS3 from the food. So first thing is we have to correct the methionine. So we have to check our food, and there should be a sufficient protein. Protein is required for many processes, and one of the processes we need methionine from the diet. Many of my patients, they are actually taking an PPI, proton pump inhibitor, or any kind of antacid, and they have a low stomach acid. By the way, you know that low stomach acid is problematic for protein absorption. But another functions of this stomach acid is it increases the binding of intrinsic factor to your B12 in stomach itself. Just think about that your patient is having a low stomach acid or they are taking antacid. With the antacid, if they are taking a B12 supplement, what will happen? That B12 cannot bind to the intrinsic factor, and if it cannot bind to the intrinsic factor, your body cannot absorb that B12 from the terminal ileum. At the same time, you have to correct the stomach acid. Make sure you understand the other pathway of acid production and synthesis. Apart from these things, as I said you, we need to increase the folate from the diet. You can increase the fruits, you can increase the vegetables, and that's how you can increase your methyl donor from the food. Two important nutrients that play a key role in methylation process is magnesium and vitamin D. So same time, if you are deficient in vitamin D, correct the vitamin D. And many speakers had talked about the vitamin D earlier. Magnesium is very important. That will help in methylation process. So this is how you're going to correct your food, how you're going to correct your lifestyle and increase methyl donor from the food. When we talk about nutrition, like what kind of nutrition, and as I mentioned you, all the B vitamins should be taken in a combination because they work in synergy. But you really need to understand about the inactive vitamin and active form of vitamin. As I mentioned you, folic acid is not vitamin B9, it's just a synthetic version. You also need to understand cyanocobalamin versus methylcobalamin. You need to understand about the pyridoxine phosphate versus pyridoxine 5-phosphate. You have to understand about the active forms of B2. So really we have to work and think that all these vitamins and minerals, they should be in a combination. So if you want to optimize, and really if somebody having a methylation defect, as I mentioned you, they might be having an heterozygous or homozygous in genes, they really need supplementation because the requirement of methyl donor is higher compared to somebody who doesn't have any methylation defect. So you need to increase the 5-tetrahydromethyl that is active forms. You need to increase the methyl B12. You need to increase iboflavin 5-phosphate. You need to increase pyridoxine 5-phosphate. Another important thing is TMG. TMG is basically trimethylglycine. It's a glycine that is attached to methyl, and 3 methyl donors are there. three CH3 group attached with glycine. And by the way, I told you from the homocysteine, you create the you create the cysteine, and cysteine is not enough to create the glutathione. So we need this vitamin in a combination to increase methylation support, and that's what we formulated as a methyl homocysteine defense, and that will help in many of my patients. We had seen patients, they're coming within more than 20 of homocysteine, and within 2 months they come to less than 12, and whatever the dietary deficiencies they have, they will control dietary deficiency by increasing their methyl donor over here. With that things, thank you very much, and you can go and check vitamin.in for more detail about the methylation and methylation factor we have. Yes, thank you very much, Dr. Karishma. We are open
Yeah.
for questions.
Thank you so much, sir, for an informative session on methylation. There are definitely some of the questions that would interest the audience as well. My B12 stays always high in spite of stopping the supplements. A genuine answer to the same?
Okay, so when you look at the chart, you need to understand that we have a two testing in a lab. One is we check a cyanocobalamin, that is only B12. So if something is written like a B12, that means the lab is checking your cyanocobalamin. It's not an active forms. There are separate tests available that we call a active vitamin B12. Or if you want to do organic acid testing, then methyl malonic acid is a better test to see your status. And by the way, if you want to do one testing to find out your B12 and your functional B12, functional folate, is check your homocysteine. If your homocysteine level is optimal, means you don't have to worry about the B12, you don't have to worry about your folate is always in optimal range, right? So make sure you understand the basic biochemistry and basic lab test. Check your homocysteine, because nobody, conventional doctor will not check the homocysteine level. First test I would recommend for anybody who is working in any chronic condition is homocysteine.
Thank you so much, sir. What is the relation of methylation with pregnancy and Alzheimer's? I guess you would be interested to answer this.
Yes, I am interested in Alzheimer's. See, I had mentioned you, Alzheimer is a chronic neurodegenerative process where you're going to increase amyloid in Alzheimer. By the way, I am researching on Alzheimer disease, and may you heard that my mother has diagnosed with Alzheimer disease and she died very young. So my patient started from there, where I'm researching on Alzheimer disease. There are many factors responsible, and new therapy is coming for Alzheimer disease. Out of methylation, one of the things I would like to give you some idea about. I'm working on methylene blue. The one promising molecule already we are using in conventional medicine, but that is also helping in many of patients Alzheimer disease. But coming to methylation, methylation is very central process, and we have to optimize methylation so our neurotransmitter and our nerve cells work better. Pregnancy, of course, you go to any gynecologist and pregnant patient go, they prescribe the three things common. One is iron, but always they prescribe the inactive iron, parasulfate. They prescribe the B12 and folate. Again, they're prescribing wrong B12 and folate. They prescribe the folic acid and cyanocobalamin. Now just imagine one patient who walk in your clinic with the pregnancy who has a MTHFR homozygous. What will happen in that case?
The folic acid will increase toxicity of folic.
Exactly. So folic acid going to bind to the receptor, even if you have active folate available in the cell, it doesn't allow to work. Just think about the active versus inactive, the active employee versus inactive employee who is sleeping at the receptor site. So folic acid is like that. If you cannot convert folic acid into the active folate because you have MTHFR defect, so this folic acid going to bind to that receptor and occupy the receptor, so it doesn't allow the active folate to work even.
Yeah.
So
And at times
yeah,
we have seen in the clinics as well that it is the leading cause for miscarriages and
yes,
pregnancy failures, IUI, IVF failures.
yes.
Yes, so
So
please keep
everybody
it
know
on.
about folic acid and folic cycle is important for initial 3 months, but that's B9, not the folic acid.
Of the
You
folic
can
acid.
understand.
Yes, thank you so much for the insightful session. We have received a lot of feedbacks as well. There are no more questions as such, but there's just one question if you can just answer in the chat box. Thank you so
Yeah,
much, sir.
sure. Thank you.
Thank you.
You need glycine